Okay, so today we're looking at assignment 4, research methods, and assignment 4 is all about quantitative data analysis techniques. So in previous assignments we've collected data, and this is looking how we use that data to make some sense and interpret it. And this first video actually just looks at some fundamentals associated with quantitative data analysis. And I'm just going to show you the assignment brief before we move on and look at the actual content. So scrolling through the mark sheet to start with and the student review, here's the front cover with the image, and just this is the bit I wanted. So this just shows you that there are several different tasks associated with this assignment, and there are a couple of different videos, 5 different videos. I think I've put these 2 on the same video at the end, and we're looking at task 1 just within this video, so it's just the basics for this type of research. And this data will be, or your work will be, written up inside the assignment brief, and here you can see there are headings specifically for you. You just obviously, as normal, type and click in the text box there, so it's all pretty straightforward in the assignment brief. So breaking down, here's our unit spec. We will work through each of these sort of subtopics. And the whole assignment is actually only a pass and merit criteria. Describe 2 contrasting quantitative data analysis techniques, so 2 different types of data analysis, and then justify using those tests for certain data that you've collected, and you will do that at the end of the task. So your final couple of tasks are using real data that you'll have collected. And the assignment scenario, all based around our British gymnastics athletes, and you're going to employ a range of research methods and tests collecting fitness data. So this is our quantitative numerical data for fitness tests. So task 1. This video will go through, I think, 5 little bits. First of all, we'll very briefly recap quantitative data characteristics. You'll look at the 2 types of tests that you might, or types of research tests that you might do. We'll look at hypotheses, sampling and populations, and variables that you might need to control in order to make your research and your data collection much more effective. So, quantitative research characteristics. Just put the video on pause and jot down, if you can, 3 quantitative research characteristics. What does it mean to do quantitative research? So hopefully you'll have put down something similar to this. So very structured research, very structured data collection, and it's fundamentally about numbers rather than words or meaning. It's factual data, numerical data. Less detail in that respect on attitudes, feelings, why, the in-depth, rich data that you get from qualitative study. And with this sort of research, you tend to need more numbers in your sample, so a larger sample size, because it has to represent a population of people, a certain group of people. These types of research or studies should be replicable, and that means that you could repeat it. The methodology is such that you could repeat it. Somebody could read your method and do it themselves. And the data analysis itself is fairly objective, so it's not about you interpreting people's feelings. It's factual data that you put into a test and you get a result, much more objective. Okay, so the 2 sort of categories of quantitative data research that you can do would be relationships or correlations, and tests for difference. So I've tried to use these images here to sort of symbolize relationships and then differences. So relationships actually look at how one variable affects another variable, or is there a correlation, is there a link, is there a relationship between 2 variables? And this example here might help you understand. Is there a correlation between work intensity and heart rate? Now, I hope you're all thinking, well, yes, there probably is, in that if you work harder, your heart rate goes up. So that would suggest that there is a correlation between those 2 variables, work intensity and heart rate. Differences test the effect of something on a group or an individual. So again, this example, is there a difference between male and female heart rate values doing a 2-mile run on a treadmill at the same pace? So if we had a sample of 20, 10 males, 10 females, they all did the same activity, 2 miles on a treadmill, running at exactly the same pace, would there be a difference between the heart rate values of males and the heart rate values of females? And we would set up that test, and we would collect that data, and we would put that data into a statistical test and see if there was a sizable difference between those two sets of values. So you may have used this phrase or this word with perhaps some understanding of what it means. Now, quantitative data looks at you have sort of a broad starting point, a research topic, and from your interest or your topic area you determine a research question, something that you want to test, and you phrase that in the form of a hypothesis. This is a testable statement. So to look at the relationship between work intensity and heart rate, you're going to look at that, and is there or isn't there a relationship? Is there or isn't there difference between heart rate values of males and females? You then collect evidence or collect data to test the hypothesis. So quantitative data analysis is very much about testing the hypothesis, the question that you have raised, as it suggests there. Now, the way you write your hypothesis matters, and there is some sort of typical phraseology or a way that you're supposed to write your hypothesis. And there are actually 3 different kinds of hypotheses. First one is possibly the most important one here, the null hypothesis. Now, the null hypothesis is the hypothesis that you are actually testing. You are testing, so the null states there will be no significant difference between, let's say, male and female heart rates during a 2-mile run at 10 kilometers an hour. Now you collect your data, you check it in the statistical test, you get a result, and once you get that result you will relate back to this null, and you will say you accept the null if there wasn't a difference, or you reject the null if there is a difference. So it's all about testing the null. So if we were using our other example, you might say, is there a significant correlation between heart rate and work intensity? And you would collect your data, put it in a statistical test. If there was a relationship, you would reject the null, because the null says there won't be a relationship, a significant relationship. So whatever your results come out as, you accept or reject the null. So the null is the key hypothesis. You also often write an alternate hypothesis, and this is the flip, the reverse of the null. So the alternate says there will be a significant difference or correlation. So the null is there's no, perhaps you can remember it, null, no. And the alternate says there will be. So if you're rejecting the null, you're going to be accepting the alternate. They're kind of the opposite. And then in some cases that you might write what we call a directional hypothesis. Now this has got this is more specific than the alternate. The alternate says there will be a significant difference between males and female results during a VO2 max test. This is still vague, you see, this is what we call two-tailed. This does not say or commit to males are going to be higher or females are going to be higher or better, whereas the directional does. So the directional suggests that there will be a significant difference, and that males will have a higher VO2 max value, let's say. So it kind of hedges its bets to one direction, whereas the alternate leaves it open. It says there will be a difference but doesn't stipulate in which way there'll be a difference. However, in our case, we'll probably not be writing or talking about directional hypothesis. We'll be focusing on the null, and we'll include the alternate in there. So it's all about the null. So our process so far, we collect data to test the hypothesis. We put our data into statistical tests, which we look at in a later video. We get the results, and based on the results, we either accept the null and reject the alternate, or reject the null, and accept the alternate. So sampling and populations then. A population basically talks about the big scale group of people that you are interested in researching about. So it could be you're interested in researching males 20 to 25 years old who play football, and you want to do a bit of study to look at some kind of effect on their participation. They would be the population, and it would be really important then that the sample you collect represents that group of people. So if you wanted to look at males 20 to 25 who play football, what you need to do is select a sample of subjects who are male between 20 to 25 years old and who play football, rather than females, rather than younger people, rather than older people. So this sample has got to represent your population. So you can see here with the colours here that the green, the sort of dark blue, the purple, the sample here represents the group of people you're interested in researching. So ideally we have what we call a representative sample, where you randomly select your subjects to be part of your project, so there's no biasing in terms of who you choose or who you end up having. Often though, for example at universities, the students will be the sample because they volunteered to take part in somebody else's research, and actually we're likely to use what we call opportunity sampling, which involves just because your class is available to you, they'll help you out just purely by opportunity. So unfortunately the way we sample is not likely to be randomized, it's likely to be non-representative sampling. Okay, lastly we need to look at variables, and the two most important variables are the independent variable and the dependent variable. Now in this example, does work intensity affect heart rate? The independent variable is the variable that you manipulate, you will change to see if it has an effect on the dependent variable, in this case heart rate. So the independent variable is the one that you manipulate, and the dependent variable is the one that you measure to see if the IV has had any effect or made any changes. Now you'll also need to be aware of all the other variables that you need to control, because we just want the IV to affect the DV. If there are other variables that might affect the DV, these are called extraneous variables. So many of them you can control, but sometimes it could be that you can't control or you didn't expect something to happen and that affected the DV, and those are called confounding variables, ones that you couldn't control. Situational variables, things commonly these will happen if the noise is occurring, which might affect the DV, or different temperatures that you didn't anticipate. Situational variables. Participant variables, somebody might come in a bad mood for one of your assessments. You can't necessarily control that. That's a variable you need to acknowledge in your write-up. Demand characteristics, so if people know they're part of a piece of research, it's possible they might behave differently just because they know they're being tested in some way. Sometimes people respond differently or change their behaviour because they think it makes them look better. It's socially desirable for them to behave a certain way. So that's called responder bias. You might change your interpretation or what you see or what you hear because you know you want to hear certain things or interpret things in a certain way to make your research work. So that might be called researcher bias. And order effect, this is quite an important one that you possibly might need to be aware of. If, for example, you're measuring free throws with and without an audience, if all of your subjects do the without and then all of your subjects do the with, it's possible that they may have practiced by the time they get to the with and be a bit better. Now that practice may obviously affect their results and make them look better. So you'd need to think about changing and mixing up the order that they do their experiments. So, in summary, task 1, we've very quickly gone through a recap of what quantitative characteristics are for data analysis, correlations and differences, hypotheses, sampling and populations, and finally variables. And again, this will be written up inside your assignment brief for task 1.